Hello.
Hello.
Oh, so it's exactly 1 now. Shall we start?
So, Mimi, all your colleagues are fine. Huh? I can see Dr. Sangi and,
Yes, yes. Everyone is here, sir.
and Professor Tulanga. He is,
Mm.
yeah, in the college. And of course, Dr. Ralte is also online. Yeah. Hello, hello. Hello, madam. Yeah, hello, all of you. Yeah. Yeah.
Okay.
Yeah. Dr.
Let's
Tommy
start.
is missing. Yeah.
I think he has some signal problem.
Oh, okay. Okay. Okay.
He'll be here again soon.
Yeah.
So,
Yeah.
sir, shall we start? It's exactly 1 now.
Yes, I'm ready. Mimi.
Okay. So, good afternoon, everyone. Now we shall start our program. My name is Dr. Remsiami Shaser, and I will be your host for today. On behalf of Government Sertip College, I thank you all for participating and giving us your valuable time to attend this 5-day international webinar with the theme Science and Technology Mitigating Current Challenges. I would just like to highlight a few of the objectives of this webinar. It is to uh, help understand what science and technology is doing in times of crisis hindering human welfare. So our esteemed resource persons are from various fields of science and technology, and we have specifically requested them to provide a general presentation so that participants from various backgrounds can benefit from it. We hope that this webinar will be fruitful and disclose an understanding to everyone. So today we have here with us Pusi Ladintuanga, an associate professor and head in the Department of Economics, to welcome the participants. He is one of the most enthusiastic teacher in our college, and he is always a step ahead when it comes to activities related to advancement for the college. It is an honor to have him here to speak for us today. So now I request Sir to deliver a welcome speech.
Good afternoon, everyone. In the 5 days international webinar organized by the Department of Botany for College on the theme of Science and Technology Mitigating Current Challenges, I extend a warm welcome to all resource persons and participants. In our present situation of COVID-19 pandemic, the world suffers various problems and faces various challenges. Due to COVID-19 pandemic, many producing sectors cannot produce their normal output. Many hospitals cannot treat normal patients as before. We cannot have normal classes in our institution. Millions of people lost their jobs and becomes unemployed. So that we have such and such problems nowadays. Therefore, we are facing this problem in our society and even in the whole, so that the world have challenges. The challenge which should mitigate our suffering. I hope that science is and technology is one of the most important weapons, most important tools in order to face our challenges. Our theme today in our work is very appropriate, as I think. I appreciate it. Once again, I heartily welcome all the resource persons and participants. And we are really grateful as you spare your valuable time for this webinar. We are proud to have this webinar with you. I hope through this webinar, all participants shall more equipped, well equipped, in order to mitigate our current challenges. Thank you.
Thank you so much, sir. So let us now proceed with our program. Before I call out our first speaker, here's an information to all the participants. If you have any question you want to ask the resource person based on the talk, you can drop your question in the Q&A box down below during his presentation, and he may answer your question after the talk. And we will be providing a feedback form which will be mailed to all the present participants today. I request you all to kindly fill up the feedback form for today and for the coming 4 more days. So moving on to our program, I will now call upon our resource person for today. I'm very glad to introduce to you the first speaker for this event, Professor Senthil Kumar. He is a professor in the Department of Biotechnology, Mizoram University, and he has been working in Mizoram University since 2007. He is among the few faculty members who established the department from scratch, and they provided MSc, MPhil, and PhD courses. He is a very skilled teacher as well as an efficient administrator. He has 18 years of research experience and 18 years of teaching postgraduate students, and many years of guiding PhD scholars. Under his guidance, 12 PhD scholars and 10 MPhil scholars have been awarded their degree. I can go on and on reading out Sir's biodata, but let me not take up his valuable time anymore since we are very eager to hear his talk. I now request our speaker to begin his presentation, and I would like to take this opportunity to thank him once again for accepting our invitation. So, sir, the time is yours.
Yeah. So, thank you very much, Mimi. So, for me, it's a great pleasure to be here among all of you. And I thank the government's CHIP College, the principal, the faculty, the staff, and the students for this great opportunity. So, it is when Mimi approached me, so I was happy that yes, fine, I'll be able to address all of you. And as she expressed, I will try to not go highly scientific. I will try to keep it as simple as possible in terms of science from a perspective of arts faculties and things like that. So let me move on to the topic, and I once again thank the government CHIP College for this wonderful opportunity. So I'll share my screen now. Mimi, the screen sharing should be enabled for me. Yeah.
Okay. I think our technicians will be dealing with that.
Yeah, yeah. Yes. Okay. Now it's okay. Fine. Is my slides visible?
Yes, sir. It's visible.
Okay. Okay. Fine. Fine. So let me continue. So the topic for today discussion is on the intervention of science and technology for human health. And so I have divided my talk into 3 basic aspects. One is I'm going to introduce maybe a little bit of biology for non-biology participants. And then I'll move on to the kind of science that we have been doing in Mizoram University, studying cancer and TB, diabetes, etc. So I'm just going to present one slide on various aspects just to get a feel of what technology can do for us in terms of human health. And the last slide, I'll be covering upon the COVID status, how science has understood COVID and what would be our possible future. So we could have a discussion upon that during the question and answer sessions. Yeah. So I start with this one very basic picture or image, which talks about a human cell, the different components that are involved in it, and for example, inside the nucleus, right, we find the chromosomes. So the chromosomes consist of the DNA segments, which are further split out into genes. And the DNA is basically made up of 4 nucleotides, A, T, G, and C. So this is what we are going to cover maximum today, that how this changes in adenine, guanine, or cytosine, thymine, the 4 nucleotides that forms the basis of our genes and DNA, is virtually helping the science in terms of human health. So some basics again, like the central, what we call it as central dogma of life. The DNA or the genes, it codes or transcribes for RNA, and this RNA translates into proteins. So proteins are basically the soldiers which bring about certain phenotypic features or morphological characters. But the genetic background is stored in the DNA, and from a chromosome, when we unwind a chromosome, we could split into the number of genes present. So humans, approximately, we have 25,000 genes in a span of the 46 chromosomes or 23 pairs of chromosomes. So these are again some of the basic definitions. So when we say a human genome, we talk about all the genetic material present in a cell, and the study of the genomes, we call it as genomics. So the genes which consists of these 4 molecules, A, B, and C, can have variants. So there can be present instead of A, there can be a C present instead of G. So these 4 molecules can interchange among themselves, which finally leads to the genetic variations that what we observe. Right. So in the evolutionary pattern, basically, this is an evolutionary time scale. So we have a wild type genotype here, a genome, a gene sequence like TG, AG. So over time, particular can reproduce and have two. Yes.
Sir, let me just interrupt. I think one of our panelists is not has not muted his Zoom, so
Yes,
we will just wait a second. We are calling him, sir.
Yes, no problem. Okay, I think now it's fine. Yeah.
Yes, sir. Okay, sir. Please proceed.
Okay, thank you. So this genes is what gives us the hereditary basis of life. So basically, these 4 molecules which are found in our chromosomes, in the genes, in the genome, get transmitted from one generation to the other generation. So this is an evolutionary time scale what we see here. So this is a wild type individual. So this individual reproduces and has 2 offsprings. So one individual has exactly the same arrangement of this bases as we find in the wild type, whereas the second individual has 1 mutation or a variant from G to A. So this we call it as a mutation, and this keeps on proceeding over time, over the evolutionary time scale. And this individual again has 1 more mutation from the parental type. There is a second mutation from T to G. So similarly, it keeps on going, and we have 1 lineage of population here, and the other lineage goes on here. So when these 2 lineages are not able to reproduce among themselves, the individuals that are belonging to these 2 lineages, then a new species is created or has evolved. So basically, this type of mutations are very common in lower organisms, very frequent. It is very spontaneous. So that is why we have a lot of species in butterflies, in bacteria. Whereas as we go higher and higher, we have very less individuals because the rate of these kind of mutations and variants accumulations are less. So this type of mutations can be harmful or can be beneficial. So beneficial in the sense it can help us to adapt to a particular environment, to a particular lifestyle, to remain healthy and to live and survive. But it can be also harmful in terms of diseases, right? So this is where the principle of survival of the fittest, right, by Charles Darwin also plays a major role. So when we talk about human disease, like for example, in the state of Mizoram as well, cancer has been a very predominant disease in the population, and many of the cancers are occurring at a very higher rate. So when in a normal, how does this cancer lead to is because of, about 10% of the cancers are genetic in origin. So when we say genetic, there can be an accumulation of series of mutations. So how does these mutations lead to cancer? Is from a normal cell, when it divides, when it replicates, can actually a few of the cells among the large healthy cells can acquire one mutation. So one base change from G to A or T to C or C to A or C to G, whatever be it. So this in a small population of cells, in a less number of cells. So when these cells keep on dividing, it might acquire a second mutation, followed by a third mutation, a fourth mutation, and later it may turn into a malignant cell. So this type of mutations can occur because of a lot of physical factors, a lot of chemical factors, environmental factors, lifestyle, food habits, and so on and so forth. But among this, 10% of this type of mutations are also genetic in origin, in the sense it is being inherited from the parents. So the remaining approximately 90% can be because of, these are the various risk factors. So science has basically helped us to understand some of those mutations. So that's what we are going to talk in the next few slides. But here, science also has helped us to understand the epidemiological and life or demographic factors, right, that can lead to various diseases, including cancer. For example, unhealthy lifestyles, poor diet, like smoking or alcohol or lack of physical activity, environmental factors like infectious diseases, in the case of cancer also, there has been a lot of viruses and bacteria that are involved in causing the disease. It can be because of radiation. It can be because of family history as well. So that is the reason why this Human Genome Project was started approximately 2 decades before. And today we have a full draft of the human genome. We have a reference genome for the world population. And among the Indian populations also, there has been a lot of genome projects. So genomics is one branch of science that has contributed largely to different branches of biology, different branches of arts, different branches of technology as well. So under this Human Genome Project, one aim of this project was to, people believe, the scientists believe that it is going to revolutionize medicine, and it is going to explain every base of our DNA, the 4 bases, A, T, G, or C. So the large medical genetic focus was to identify variations in the genome that is disease causing. So in the sense, human genome also, what are the variants that can lead to a disease? In the sense, an infectious disease, for example, like coronavirus. So what can be the variants that are present in its genome that can make the virus more virulent or more suppressive in nature? Or can it be more symptomatic, or will it remain asymptomatic, right? So a lot of research is now into these processes as well. The second focus was on to determine how individual genes play a role in health and disease in terms of human genome. So these are the 23 pairs of chromosomes. It is a karyotype that what we see here. So if we sequence all the 3 billion base pairs that occurs in the human genome, in a regular newspaper size paper, it could fill 1,51,910 pages of A, T, G, and C. So that much of information is stored in every nucleus, that is what makes life very interesting to understand, and that is also has made life for biologists to be understood in a very less amount till today, even though the technologies are available. So another interesting aspect of the human genome is about 99.9% of the nucleotide base arrangements, these 4 bases, are exactly the same in all the people. So whether we are an African, American, European, or an Asian, the base changes almost more than 99%, right? Remains exactly the same. So about 0.1 or 0.2, maximum up to a level of 0.3% of these among the 3 billion bases vary. Means variants are accumulating in those genomes, and less than 2% of the genome codes for proteins. In the sense, among the 25,000 genes that are present, like among the whole amount of the human genome that are present, less than 2% codes for the genome codes for the proteins. So remaining 98% codes for other small non-coding molecules, which actually regulates the function of this 2% of the genes. So this is the major human races that occur across the world. And these races, even though with more than 99% of the genome, the 0.1 or 0.2% of variance in this genome causes such kind of diversity in terms of morphology, physical appearance, culture, food, lifestyle habits. So people get adapted to the environment where they live in, and they survive according to the environmental strategies. So the genes interact with the environment to produce a particular phenotype. So what is the use of studying this kind of structural variations is because it is common in the normal human genomes, right? So we find a lot of variance even among brothers, even among sisters, even within a particular population, even within a particular continent. Among the world population also, you find a lot of variance that can exist, which is a major cause of the phenotypic variations, the physical appearance of the people. But more importantly, it is common in many of the diseases like that are hereditary in nature, like cancer or diabetes. People these days even attribute genetic variants to a disease like tuberculosis, like how variants are in the genes present in the humans can lead a person to have the disease. Whether some people, even if they come in contact with mycobacterium, they may not exhibit the disease, or it may remain asymptomatic only because their genes doesn't have those mutations or variants. Now it is also showing up in many of the rare diseases that exist in the families. Neurodegenerative diseases like Alzheimer's, autism, Rett syndrome, many of these diseases which were not known in the population, but it runs in the families. So these kind of rare diseases also, the gene variations can be attributed to. So according to some of the findings, people say that 5% of live born babies have congenital disorders which have a genetic component in their etiology. And most of the common diseases of our later life, after the age of 45, 50, 60 years, they have a genetic component. So when a child is born, after a few years, if the genome is sequenced, and if we read into the variants that are present across the genes or the entire genome, we could easily predict, or we could have some risk factors that could, some of the diseases that the individual or the person may face even in their later part of their life. So that's the advantage of studying genetics, human disease genomics. And these kind of disorders have been responsible for an immense amount of suffering, reduced quality of life, and shortened life expectancy and distress, etc. So this is one slide which just gives you an example. This is from the literature, which talks about how the mitochondrial gene mutation. So mitochondria we inherit from our mother. It is maternal in inheritance. Mitochondria has about 37 genes. So these are different from the chromosomal segments that we discussed so far. But they also play a very important role in health and disease. For example, this is the name of the gene ND1. So in a normal healthy individual it is A, whereas in a tumor sample it is G. And these are found in certain populations only. For the cancer patients, these variants in this particular gene have been found or have been discovered only in certain populations. Similarly, in the same gene, but another location, there is a transition from G to A, from healthy individual, a cancer patient has A in that place, right? So this is also specific to certain populations. So like that, almost in many of the genes among the mitochondria, among the 37 genes also, people have studied not only for cancer, but also for various other diseases. This is with respect to breast cancer, the BRCA1, BRCA2 genes, the most popular gene that has been studied for breast cancer. This is chromosomal gene found in the nuclear genome. So for the same disease, among different populations, there has been different types of mutations or variants or structural variations that have been found in these two genes, and which are or also which involve in the process of carcinogenesis. So that is the importance of especially the genomics part, and at a broader sense, application of science and technology for understanding the cause of the human disease, as well as not only for the cause, it is also very useful for early diagnosis and late prognosis as well, up to late prognosis. That means it can even help a clinician, a doctor, an oncologist to decide what kind of treatment to be given for a particular mutation that is present in a particular patient. So this is one another slide, an example slide, which tells you about these variations that are found at the DNA level in different individuals. How does it influence our treatment strategies? So one size does not fit all. So this slide explains you the percentage of the patient population for which a particular drug in a class is ineffective. For example, if we take the case of diabetes drug, wherein even metformin is the most popular drug that is given for the diabetic patients, approximately 43% of the patients will not get benefited because of metformin. Only the remaining, this is a world average, right? So it can differ from population to population, plus or minus, it can be few percentages. So similarly for cancer drugs also, only 25% of the patients get proper or exact or what we call it as a perfect treatment, and they get cured. 75% of the cancer patients, they don't get benefit from the kind of drug or treatment they are put into because of the gene variants that are present in their genomes. So that was my basic, the part one of my talk, where I wanted to explain what is the importance of looking into this kind of genetic variations that are present in the human genome, and how does it contribute to our understanding of the disease, and furthermore for the treatment of the disease. And one important concept that we need to take home message from this is every population will have the variants differently, and every individual may also have a new variant that is not even present in a population. So from here, I'm going to move on to some of the next few slides is going to talk about our research that we have been doing in Mizoram University for quite a decade now, and what kind of findings we have got. So I'm going to be very brief. I'm not going to explain much deep science into this. So we work on gastric cancer for the last 5-6 years because gastric cancer is one of the cancer, unfortunately, in which the Mizoram state leads in the country among men, right? So we took up this work, and we have been sponsored by DBT for that. So this is our collaborator from NIBMG Calcutta, Dr. Arindam, and these are my students who worked in the gastric cancer project, and of course still continuing as well. So the germline mutations is what is basically hereditary in nature, in the sense the blood DNA will have these mutations which can be transmitted to the next generation. So in that perspective, we sequenced the blood DNA from patients we got the samples from Mizoram State Cancer Institute, Civil Hospital Lysol, and we sequenced the genes, wherein we found some of the frequently mutated genes that are found in the gastric cancer patients among Mizo population, which are not found in healthy individuals in Mizoram or among Mizo population. So MAP3 kinase, MAP3K4 is one of the gene which is highly mutated to the tune of 92%, followed by the different genes that are involved in the process of carcinogenesis. And this can be used as an indicator. For example, another gene, FAT4, which is also to the tune of about 21% in the gastric cancer patients, was found to be significant, and which can be used as an indicator of early stage gastric cancer, like gastric cancer at a very early stage of the cancer disease itself. And MACF1 gene mutation, another gene, was found to be significant with advanced stage and poor survival. Even after surgery and treatment, the mutations of this gene makes a patient to have a poor survival time, right, or frequency, indicating the need for developing targeted drugs for the population. So all these genes that we have listed here could be an underlying predispositional factor. So the reasons why gastric cancer is very high in the state of Mizoram could be explained by mutations present in this gene, and to a great extent, the patients could benefit from the kind of treatment that could be done to target these genes and the proteins coming out of these genes. So we also did somatic mutations. So somatic mutations are acquired mutations, not through hereditary, but because of smoking, because of alcohol, or because of food habits, consumption of smoked food, etc. So we studied a questionnaire-based study wherein we found tuibur consumption and smoking habit also are very important risk factors for gastric cancer. And among that gene study, TP53 gene, the tumor suppressor gene mutation is found to be highly significant. There is a general knowledge question which says that why do elephants don't get cancer? Right? Many animals get cancer, including dogs and other animals. But why do elephants don't get cancer is because of the fact that elephants have multiple copies of TP53 gene. So even if a single copy of the gene is mutated or some variants are present, the elephant is able to match it with other copies of the genes. So it is multiple copies of the genes of TP53 are found in elephants, so they don't get cancer. So TP53 gene is a very, very important gene in many of the cancer pathways among the world population, right? So we also did a whole mitochondrial genome sequencing apart from these nuclear genes, and we found some of the genes that are involved in complex, out of the 37 genes, about 7-8 genes belonging to complex 1 and complex 4 mutations also played an important role in gastric cancer. So we also work on breast cancer. So this is our collaborator from Mumbai, Tata Hospital Research Wing, ACTRAC, Dr. Ashok Verma. And Doris was my student, and Dr. Maumi, she's a pathologist in the civil with, she's also a researcher now. So some of the risk factors with a study of more than 400, 500 breast cancer patients, we found these are some of the risk factors in Mizo population, which are significant in terms of the disease. So for example, consumption of saum, the fermented fat, pork fat, consumption of smoked food, right, kidney consumption, or first or second degree breast relatives are hereditary, right? The cancer runs in the family, right? So, or even having other inheritable diseases, lack of exercise, or duration of lactation, or age at menopause, some of these factors. Even interestingly, we also found that in Mizoram, the consumption of fish is also a significant risk factor. Fish is otherwise thought to be very healthy food, rich in protein, but unfortunately, because of the presence of lot of preservatives like injection of formalin or formaldehyde into the fish sources makes the fish toxic, and consumption of fish, the fish that is imported from other states, from South or from North India, is found to be very big risk factor for breast cancer. So coming to the genomics part of it, BRCA genes we already discussed, BRCA. Angelina Jolie made this gene very popular among the world surgeons and clinicians. So we found two important risk factors here. In this BRCA1 gene, when we sequenced, we found an alteration from T to C. T is in the healthy women, and C is in the breast cancer patients. Similarly, another alteration in the same gene in a different location, C to T. So this is the sequencing profile, the graph that you see here. So these genes, like for example, what is mentioned here at this level is considered to confer cancer susceptibility. And one of these mutation that we found here is also found to be novel, in the sense it is not been reported so far in any of the other world population or patients. So this type of novel mutation finding is very, very important and significant in terms of identification of the mutation causing factors and also having a genetic counseling. So these two other variants, like the case of what we find here, might provide a potential diagnostic or biomarker, prognostic biomarker for the population. So how do we identify, in the sense, the individuals can get sequenced at an early age, at the age of 10 years, 15 years. And if there are any variants present in them, then possibly they could adapt a healthy lifestyle. They could possibly, undergo kind of a counseling in which they will be able to avoid the disease, or at least instead of getting cancer at the age of 40, 50 years, they can postpone it to 70 or 80 years just by genetic counseling, right? By understanding this for diagnosis or even for treatment strategies. So even in a mitochondrial gene, we found some of these genes along with the nuclear gene, what is listed here, were found to be druggable genes. So genes which can be treated. So a mutation present in these genes, if properly identified and treated, the patient can get cured of the disease. So these are some of our publications that we had made in the recent years from our study. So we also work on diabetes in Mizo population. So this is our collaborator from Jammu, Savarkar, and my student here, Freda. Here, for the case of diabetes, we found MACF1 gene variant with this RSID is found to be significantly associated with type 2 diabetes in Mizo population. And the heteroplasmic variations that are found in this mitochondrial gene is found in the 36% of the samples. Some of the lifestyle habits like consumption of saum, smoked food or smoked meat, excess salt consumption, tuibur, kuwa, sada consumption also were found to be involved. So main factor, the driving force might be saum consumption, followed by the other factors as confounding factors which support the disease, diabetes. And these are some of the gene lists that have been identified with the variants with the RSID given below, where we've found that these variants are found in the diabetic patients, and this is involved in the pathway that leads to the disease. So these are some of our publications that we did in the last few years with respect to diabetes, gene variants, and lifestyle and food habits in Mizo population. So we collaborate with the Gastric Cancer Hospital in Silchar, Dr. Ravi Kannan and his team Rajiv, and this is my student Alitika. So we work on colorectal cancer using the stem cell markers. So here we found ALDH1 gene, the aldehyde dehydrogenase gene, to be overexpression of this gene associated with poor survival in colon cancer and the colorectal cancer patients. And this can also correlate with the early onset of the disease, that is below the age of 40 years. This overexpression is a biomarker or an indication for the disease. So we found using next generation sequencing approaches, plenty of genes which are found to be upregulated in young age colorectal cancer patients, and followed by the late onset of the other genes which are involved in the late onset of the genes. So we did a work with one of my student, Andrew, who is also right now sitting with me. So we worked on pediatric leukemia patients. We collected the samples from Mizoram State Cancer Institute, Zemabok. And we collaborated with Dr. Jerry and Dr. Doris, along with other doctors, oncologists from there. So these are two of our collaborators, Dr. Sridhar and Dr. Vinod from IGIB, New Delhi, Institute of Genomics and Integrative Biology. So a novel FLT3 gene mutation, which had a 9 nucleotide basis, ATGC insertion, was found in one of the child patient below the age of 2 years. And various other mutations in the mitochondrial gene was also probably involved in the pathogenesis of acute myeloid leukemia. We found it to be present in across all the about 15 samples we had analyzed, children's below the age of 2 years or 3 years. So we found this type of mutation from the mitochondrial gene to be present, the T2C change, right, which are inherited from the mother basically. And 2 unreported novel variants were found in the Mizo population in these 2 genes, ND2 gene and ND6 gene in the Mizo population only. So these are all some of the technological developments that helps for the clinicians and the oncologists or the surgeons or the pathologists to actually go in for a particular type of diagnosis or for a particular type of treatment. So this figure just lists out the total number of variants that we found in the different genes in the mitochondrial genome. And this is one of our publications that we did it very recently. Another DBT sponsored a project on MDR TB, tuberculosis in Mizoram. We were looking at the drug resistance. So these are some of my postdocs, Senthil, Sarath Babu, my PhD students, Christine and Mary, along with our collaborators, Partha Majumdar, who is the president of the Indian Academy of Science now, and he is a distinguished professor of National Institute of Biomedical Genomics, Kalyani. Shandil, he owns a company in Bangalore. Dheeraj is from ICGB Delhi. Basuti is from NIBMG. So here also we have so far did a epidemiological study involving 275. We work with doctors from Falcon TB Hospital, with the state TB officers and the district TB officers. So we collected a questionnaire sample of 275 TB patients, and then we found that some of the risk factors like alcohol consumption, smoking habit, and smoked food were major risk factors. And we sequenced the patient samples to find out that these genes that have been listed here, including the toll-like receptor genes, vitamin D receptors, gene mutations, were found to be present in all the samples that we sequenced. So these are all highly significant gene variants that needs to be identified and understood, which can lead to better recovery of the patients. So Mizo population harbors 17 of those variants, which makes the population predisposed to mycobacterium infection. We also sequenced the mitochondrial genome, and we found some deleterious mutations in the mitochondrial genome. So this is our recent publication that we did from this study. So we have embarked upon whole genome sequencing of the healthy individuals as well. So in collaboration with IGIB, Institute of Genomics and Integrative Biology, with Dr. Sridhar and Dr. Vinod, the CSAR laboratory in Delhi, we are involved in these projects where we, from the Northeast side, we have contributed about 121 samples with 53 samples from Mizoram for whole genome sequencing. And we are now in the process of starting trying to identify the genetic biomarkers for different diseases in the population. So it's a most comprehensive genomic database for Indian populations, giving a good data. So apart from this, certain clinically actionable variants, what we call it as genomic precision medicine oriented technologies, are also possible using the genome sequences. Here is one example which talks about 5-fluorouracil. So this is a chemo drug invariably given to all types of cancer patients after the surgery. But there are reports which says that about 40% of the patients are susceptible to this drug, means they are sensitive. They try to have some side effects or they don't get cured because of the disease. So this 5-fluorouracil chemo drug is metabolized by a gene product, a protein. The gene name is DPYD, and the mutations in this gene can classify patients as increased risk for the drug, or highly reduced risk, or moderate risk, and things like that. So we are now applying this technology in collaboration with MSCI and Civil Hospital to find out the gene variants in DPYD gene in Mizo population, to understand whether 5-fluorouracil could be given as a drug to the patient or not. So this is one kind of diagnostic tool that we have already implemented. So this is one of our publications in Human Genomics Journal in collaboration with IGIB scientists. And in this projects, every individual is given a genome card. The Honorable Minister, Dr. Harshvardhan, is holding this card. So this genome card, the participants get a genome card, and this is like kind of an ATM card or card, but which doesn't give money, but it tells about yourself, your entire genome, the variants present in you, which type of drugs are good for you and which type of drugs are bad for you, what are the mutations that are present in your genes that might probably be a risk factor for various diseases that are prevalent in humans. So there is another project that we are also involved in through DBT New Delhi, the Genome India, which is a population-wide genome scale sequencing. So there are about more than 20 research institutions in the whole country that are involved, and we are the only university in the whole country that is involved. Mizoram University is the only university involved in that project, where we tend to sequence the whole genomes, create a genome map for the Northeast populations, create an exhaustive catalog of genetic variations, construct a reference genome for the Indian population, design genome-wide arrays for undertaking diagnosis or basic or clinical research, and have a kind of a database and a biobank. So this is because India has been grossly underrepresented in worldwide studies for genome studies. Even if we sequence our cancer patient samples from here, we need to compare it with the European population, American populations, which doesn't give us an exact picture of, it tells us approximately up to 80% or 90%, but not more than that. So we need to go deeper and find out the real, 100% of the variants, the cause of the disease, and the treatment strategies that could be made population-wise. So that's why this Genome India will be the first of its kind in the country that aims to bring together for this large-scale project to build a national genetic resource. So one another study in collaboration with our collaborator from Kanapara, the Assam Agricultural University, Professor Prabodh Bora, we studied the occurrence of Listeria species pathogen in the foods of animal origin. So we collected the chicken samples, pork samples from Mizoram and from Assam, and then we found the lot of Listeria genes that are pathogenic, those isolates which have those pathogenic variants which can cause the foodborne diseases. So we also found antibiotic resistance in those isolates of Listeria monocytogenes, so which also is a health risk factor. So now I move on to COVID-19. So as we all know, this pandemic period has been a really big difficult situation for countries across the world. So now because of application of science by scientists and researchers across the world, now we have understood this coronavirus to a great extent. For example, this is a RNA virus. The genome is RNA and it has about 30,000 bases. It is zoonotic in origin, that is transmitted from animals to humans. And we have 80,000 genomes sequenced so far across the world, which has told us many things about the spread of the disease as well. So we also know through media and other sources that the mean incubation period is 5 days for the disease. So that is why people do 14 days quarantine, or some quarantine period is also extended. So this mutation, this virus basically acquires this mutation in the genome in less than 10 days. One mutation in a year, around 360 or 370 days, it may accumulate 10 mutations. That's the mutation study that scientists have done. So approximately one mutation in less than 10 days, this virus accumulates in itself. And that is why new strains or new genotypes are spreading across the world. And according to Trevor Bedford 2020, they say that as mutations accumulate in the coronavirus genome, they allow scientists to track the spread of the COVID-19 across the world. So what they have also found is that every two infected individual, the mutation would have already, one mutation would have already come into the coronavirus genome, right? So approximately about, a period of 14 days. So the first coronavirus genome variant was identified in December 26th in China, followed by the next coronavirus genotype on January 8th, right, 2020. So within a span of 14-15 days, this the virus accumulates the mutations, so in a matter of less than 10 days. And this genetic variation in the COVID-19 coronavirus genome itself helps us to understand even the community spreading. So by sequencing the genome, we will know which variant of this genotype has come from which location of the world, and within the country or within the community also, how is it spreading? How is it moving from one variant to the other variant? And how are these variants, going into, mutating and spreading? Not only that, for contact tracing, but also for diagnostic purpose, for detection of the coronavirus using RT-PCR methods, or for antibody testing, the rapid testing kit, or so on and so forth, for vaccine development and for management of the disease. So this another interesting fact about coronaviruses, even though Mizoram had so far more than 1,000 cases, but active cases is less than 400 cases currently today. There has not been even a single death. So genetic variations in the humans, right, in the ACE2 receptor or immune-related receptors can lead to either genetic predisposition or resistance to the COVID-19 infection itself. And some of the host genes like ACE2, TMPRSS2, HLA, the immune-related genes, TLR, toll-like receptor genes, CFL, DSTN gene, MBL genes. So these are some of the genes that one needs to now study in Mizo population to understand the extent of the infectivity of the coronavirus, and also to see what type of vaccine will work for the patients from Mizoram, what type of new diagnostic methods could be developed, and how this disease can be managed. And by sequencing the coronavirus here in these samples, one can understand how this virus is spreading. Community spread or contact tracing could be much more made easier. So we are already in touch with the ZMC COVID-19 testing lab, and we are going to start these two works both on coronavirus as well as on the host genetics. So this is my last thank you slide, and I suppose I'm on time, and Mimi will not scold me for exceeding time. But before we conclude, on the left-hand panel, I have shown you all our collaborators. Dr. Jerry from MSCI, Dr. Saya, the surgeon from Civil, Dr. Eric, who is the mission director, Dr. John, the deputy superintendent and pathologist, Dr. Marchana, pathologist, Dr. Fella, who was the earlier state TB officer, Dr. Fabiola, the current DTO, Dr. Zotan Kumar, the current STO, the state TB officer, and ENT doctors, Dr. Dinga is here. Dr. Zotana is an medical oncologist in MSCI. Dr. Lily, earlier, she was the district TB officer. So we get excellent support from the clinicians. They are our very good collaborators. We collaborate with them on different diseases and different aspects of disease epidemiology as well as disease genomics. These are our collaborators externally, which I have already shown in those slides individually as well. And these are the panel of students and postdocs and the research scholars and the lab technicians like Santhya, David, Sarath Babu, Subhajit, Freda, Dr. Maomi, Christine, Payal, Doris, Deepika, Litika, Andrew, Mary, Ranjan, Zami, and Michael. Pooja, and the other are our lab technicians, and these are the earlier students and postdocs. So we thank DBT actually, Delhi, for many of the support and for the infrastructure in the department, establishment, as well as for research and the research projects. So with this, I finish my talk and I'm ready to take any questions. And I once again thank the Government Chief College, the principal, the staff, and the faculty, and the department of botany, especially for this great opportunity. And I suppose it has been a useful talk for the participants. Yeah. Thank you.
Thank you so much, sir.
Yeah. Thank you.
Yes, thank you, sir. Your presentation was very informative. And can you hear me? Am I audible? Okay.
Yes. You are audible. Yes.
Okay, sir. So I suddenly felt kind of nostalgic also to see some of my old friends, MZU friends, yeah, Senthil and David's face.
Yes.
Okay, you have been working with a vast collaboration with so many people. Your field of research is very interesting, and I can also say that you are kind of a blessing to the people of Mizoram. So
thank you. It is our duty, and it's a great pleasure to work here in the Mizoram University. It's a base for doing research. Yeah.
Okay, sir. So it's in the chat box. Also, many participants have shown their appreciation for your presentation. So there are some questions. If you are ready to answer these questions, I can read it out for you.
Yeah,
okay.
Yeah, please.
Okay, so the first question is it possible to detect cancer from DNA? Is it possible to detect cancer from DNA?
Yes. So basically, cancer is a excess growth. So you will find a tumor presence, and then at the DNA level, what we see is the mutations, the variants that can be found. The cancer cells have, they replicate in a very fast duration, unconditionally. They keep on replicating like a bacterial cell. If there is a space and if they have the nutrients, they keep on dividing. So this division, the cell cycle process itself, there has been a lot of studies that has been done to understand the progression of DNA, even cell division or DNA replication with respect to cancer. And people have found that, the cancerous cells exhibit like that kind of an activity which helps in their propagation very quickly. So mutations present or accumulating in this genes or at the DNA level also are very good early indicators for cancer to predict or to diagnose.
Okay. And a second question is, can consumption of betel nut, that is guava, be a risk factor to cancer? A risk factor of cancer?
Yes. So there are two things involved here. The betel leaf itself has some medicinal properties. The leaf is very good for consumption. People take it for digestion and other purposes. It has antimicrobial activity, antibacterial properties. But the problem lies in the kuwa, right? The betel nut. Because largely in the Northeast, people consume the raw betel nut, the unprocessed ones. So the problem lies there. So there has been one study which showed that after eating kuwa, within a span of 20 to 30 minutes, if people drink like a Coke or Pepsi, the drink which are gaseous, there are high chances that oral cancer is induced. So they have did this work in cell line study where the kuwa is treated first, and then those kind of hydrogenated drinks like Pepsi, Coke, within a span of 20 to 30 minutes, that has the property to induce cancer in those cells. So the raw unprocessed kuwa is the problem. The second problem lies in the chuna, right? The calcium. Most of the calcium comes from underground reserves of Meghalaya, where radioactive materials are also taken, right? Uranium or thorium and all is taken from a largely from Meghalaya, right? So calcium is also taken from those reserves. So there is a study which has shown that this calcium has trace amount of radioactive materials. So daily consumption or frequent consumption can induce cancer. So these are some of the problems with kuva. Otherwise, betel leaf as such, little consumption of betel leaf is healthy, basically.
Okay, that is very informative answer, sir. I think we should start considering stopping eating kuva by now.
Yeah, at least the raw unprocessed betel nut and calcium, we need to be a little more careful from the source of the calcium.
Okay. So the next question is, can we mutate our genes manually in order to combat some diseases?
Okay, so that is actually the new technology, CRISPR-Cas technology, right? So which can do gene editing. So people have been trying out in various diseases now, even with respect to human diseases. So they are trying to correct those mutations in the cell lines and then make it proper so that the disease is not induced because of the gene mutations. So it seems it will be possible in the future, right? Earlier days, there was RNA interference, RNAi technology, which can help in, kind of these mutation studies, understanding and helping in mutation or overcoming these mutations. But now CRISPR-Cas technology is a very new technology. It has been implicated, like, Andrew is telling me to tell about HIV-free in China in twin babies. Yeah, so there is a case example, right? So it is coming up. Even the CRISPR-Cas technology is a very new technology, only for the past 1-2 years, right? And it might be possible, definitely, to edit those mutations, correct those mutations in the cells.
All right. Okay. So the next question is fern, takok in Mizo, is it carcinogenic? So do you know this plant, takok?
Coke.
Have you heard about it?
Coke, I don't know. Maybe I have seen it. But one thing, what I can say is large or daily consumption of green leafy vegetables, right, is also a risk factor because it has got a lot of secondary metabolites in them. So those secondary metabolites, like amines, amine compounds, even phenolic compounds, tannin compounds, right? Accumulation can lead to cancer. So when I visited Delhi last time, IGIB, one of the scientists said that, in the Northeast, cancer is very, very high because there is large amount of consumption of green leafy vegetables. Then I was also surprised. Then I asked him, and he said amine compounds, right? They are very high in content in the green leafy vegetables. So that is also a risk factor. So even fern, this question specifically, if we can analyze, if we do, antioxidant study, and if you do phenolics, if we do phenolics, tannins, or estimation of those secondary metabolites, then probably we'll be able to say.
Okay, sir. Next question is, sir, please let me know why the breast cancers are common in female than male.
Oh, yes. Because of, the lactation process, right? The the hormonal status, the estrogen, progesterone, and HER2, right? PR, HER2, right? In any pathological state of the breast cancer, these three hormone level is checked, whether it is triple negative. Triple negative cancers, they don't have any treatment. So based on the hormones present in a female system, right? And that is also involved in the process of lactation. The basic physiology is built, right, in that fashion. So even though this is a very good question as well, all your, these are students, is it? They are asking very good questions. Yeah, excellent. So these genes, even though they are BRCA1, BRCA2 genes are present in male also, but the phenotypic expression comes along with the pathological state, the physiological state of the system, right? The hormonal status, the the conditioning, right? The need of the role of female is different, role of male is different, right? So basically the metabolic and physiological functions.
Okay. And the next question is, sir, can you please explain in brief the comparison between genomics and proteomics?
Oh, yes. Yes. So genomics, like we study the whole genome present in a cell, like be it chromosomal, be it mitochondrial, or if you take plants, then chloroplast. When we do genomics, we study the structure of the genes. Like we sequence the genes, we try to understand their gene arrangement, and we stop there, right? When we do basically proteomics, we understand the physiological role, the functional role of the product of the genes, right? The structure of the proteins, the function of the proteins, right? So this is how there are different techniques, tools, and methods for doing genomics and proteomics separately. But a combination of both is always the best, right? To understand any disease or other functions.
Okay. So there is one question that one of my friends wanted to ask. So you said that fishes that are imported is carcinogenic, right? The ones that are imported, and especially in Mizoram. So is there any research on it, or is there any scientific proof done?
Yes, yes. One of my PhD student, Doris, so during her study, she did a small experiment. She collected fish from different sources, of course, from Aizawl markets, 3-4 markets, but fish which came from Andhra, fish which came from Silchar, fish which came from Punjab, and fish that is, caught from Mizoram, right? So she had different fish varieties, and she estimated the level of formalin content in those fishes. And we found fish in Mizoram also has formalin, but at a sublethal dose, below the toxicity level. But the fish from Andhra and from other sources had 10-fold higher level of those contents, that toxicity content. In one of our, because she did a questionnaire survey of more than 275 breast cancer patients. She met them personally or through the doctors. And then we took a full questionnaire about their lifestyle and food habits. And she also did from 400 healthy Mizo individuals, right, who don't have breast cancer, women. So in her statistical analysis, consumption of fish was a risk factor. And we were surprised. That is why she analyzed the fish for formalin content. And we found the fish which was imported or basically comes from other places with the preservatives, right, has very high content, even 10-fold above the sublethal dose. And that is why maybe in our epidemiologic study, we had got a consumption of fish as a risk factor for breast cancer in Mizo women. Otherwise, fish is considered to be a very healthy food, right?
Yes, yes, right, sir. So is there any question that the panelist member want to ask?
Mimi, I hope I was not too scientific in my talk.
No, sir. It was really good. It was simple and it was very informative.
Okay, thank you. Slides were mostly prepared by Andrew. Yeah.
Okay. That's a good work.
Yeah, yeah, he is listening to you. Yeah.
Okay, I think Dr. Sidaramrata wants to ask a question. He will be our speaker for on the 4th day, that is on Thursday. He will be our speaker.
Right, right. You introduced him also on the other day, right?
Yes, sir. Yes, sir.
And how he got introduced in your life, yeah. Yeah.
Sir.
Yes.
Yeah, I really appreciate your presentation.
Oh,
and it's very informative.
Thank you.
Going to be very important for the people of Mizoram also. So you have found many important things, and based on your research, I came to understand that like majority of the lifestyle of the Mizo have to be changed, like the fooding habits and many things. So there's just two questions that I would like to ask
Yeah.
regarding you have mentioned regarding the vegetables. So you have mentioned that the continuous consumption of the green leafy vegetables can cause cancer.
Yeah.
So
High consumption. Yeah.
high consumption.
Yeah.
So can you be more particular which type of vegetables that you study or in which particular locations, so that people will be able to know this consumption of this kind of vegetable is not safe? And
Yeah.
my second question is that in Mizoram, consumption of smoked pork is very popular,
Right.
and even if we tell the people also, they won't follow. It's very difficult. So will there be any alternative like smoking of the pork? People usually use the firing of these firewoods, using of firewoods.
Yes.
So instead of using the firewoods, will there be any other alternative like using of electric heaters or something like that? So what is your view in these things? These two
Yeah,
questions.
yeah. Thank you for the questions. Yeah. So the first question, like with respect to the green leafy vegetables, we could so far do only a very small study. We have not done a very elaborate study, and this is one study where someone has to take up at a large scale and do it. Even, for example, wild edible vegetables or wild mushrooms, we have to study. So we only analyzed about 6, 7 green leafy vegetables like antrum, zonta, yeah, so some I don't get the names now. Only 5, 6, and we did a very small study, a very small essay. So which is not very conclusive actually scientifically. We have not even published it so far. So, but if somebody can do it at a very large scale, we would also be happy to collaborate and help each other and conclude something in a very big way. So this is one area of your question that needs more research. Some institute has to come into, maybe some college or university or research institute has to take up. So I don't have the answer now. Second question with respect to smoked food. Yes, you are very right. Burning of wood, the smoke creates free radicals. So free radicals means it generates the nitrous oxide and other smoke compounds, generates free radicals in the human system. And those free radicals goes and damages the DNA, and that's how cancer or many other, even for example, diabetes also, it goes and affects all the metabolism and metabolic functions. So like alternative to use of vegetables, of course, there are electric cookers, and in general, the steamed foods are the most healthier ones. The earlier lifestyles, which our ancestors followed, right, are the best one. The direct burning on the fire using firewood or other things will cause some kind of compounds that can be carcinogenic. Even meat or vegetables that is burnt, the black substance after cooking, smoked food, if the black substance is present, we should not eat it. If we consume it, it goes and causes free radicals inside the system. So in general, the boiled types are the best in terms of human health. If you are interested to know more about this, we could be in contact with one of our chemistry professor, Kumar. He is from chemistry department. He has a very good knowledge on this free radical generation and the type of the substance for cooking food and other things. Maybe we can be in touch with him. Yeah.
Okay,
I can introduce him to you, and he will explain you the full chemistry behind it.
yes,
He's
yes.
also an excellent speaker. Yeah.
Okay, sir. Thank you so much, sir.
Thank you. Thank you.
Okay. Um, I think I just want to clarify about that question, about the Takok question, that Fern. So there was some 2 years back, I think there was a confusion regarding the identification of this plant. So in Mizoram, we eat what we eat this Takok. The botanical name of it is Diplasium esculentum. So it is belonging to the family Atheriaceae. So this is not carcinogenic, and I think it is confused with another plant which looks like sakok. So that one, the botanical name is Pteridium equinellum. It is also called as bracken. So it causes stomach cancer in Japan, and I think research has been done on this plant. But I just want to clarify to the participants that it is completely different plant. So the sakok that we are eating, it is not carcinogenic, I think, so far.
Okay.
Okay, sir. Thank you so much for your time.
Thank you.
And
it was great pleasure to meet your, faculty and staff. And so, thank you for the opportunity. Yeah.
Okay, sir. Thank you, sir. It has been an honor to have you here in our event. Your participation ensured the success of our event. So we have reached the end of today's session. We hope to see you all for the coming 4 more consecutive days. And don't forget to fill up the feedback form, which you will receive in your email. So thank you, everyone.
Yeah, thank you.